This is the introduction and information for Lab 10, Mitosis and PCR. The cell cycle describes the life cycle of a single cell. And for a single cell, it goes through two major phases. The first phase is called interphase, and that's where the cell spends most of its lifetime, 90% of its lifetime, doing its normal function. The other phase is the mitotic phase, and this is the phase where the cell goes through cell division. For interphase is made up of three steps: G1, S phase, and G2. For mitosis, that phase, the cell goes through a more complex system, which will result in cell division. As I mentioned before, interphase has three stages. First is G1, and G stands for gap, so the first gap, G1. This is where the cell gets the signal to divide. And the cell will start growing in preparation to divide. Once the cell gets to a certain size, the cell will enter S phase, and S stands for synthesis. And this is the stage where the DNA is copied. So each daughter cell is going to need a copy of the genes so it can function as the parent cell functioned. Once the DNA is copied, the cell goes through G2, so gap 2 phase. And this is where protein synthesis occurs and the rest of the cell gets ready to divide. Once the cell has completed G2 phase, it will then progress to the mitotic phase, also known as mitosis. And mitosis is a type of asexual reproduction. And asexual reproduction is where one parent cell will divide to form two identical daughter cells. And these cells are identical because they have exactly the same number of chromosomes as the parent cell with exactly the same types of genes. All the cells in the human body, except for the egg and sperm, divide by mitosis. And there are 4 stages in mitosis. The first stage of mitosis is prophase. And during prophase, there are 4 different things that occur. First, the chromatin condenses into chromosomes. So the unwound DNA winds up to form visible bodies called chromosomes. Second, the nuclear envelope is going to break down. Third, the nucleolus disappears. And fourth, the mitotic spindle forms. Here is an image of interphase here. So that's interphase and then prophase. Interphase, what you see is a nice defined nuclear envelope and uncondensed DNA in the nucleus, so you have a nice even color in the nucleus. When prophase begins, this is early prophase here. This is early prophase here. This is late prophase here. What you see is a progression of these 4 different things occurring. So the chromosomes are condensing until they are fully condensed during late prophase. You also see the nuclear envelope breaking down until it's all gone at the end of late prophase. The nucleolus is going to disappear and the spindle fibers are forming. These are the spindle fibers here, and they are forming and attaching to the chromosomes. The next phases in mitosis are metaphase, anaphase, and telophase. Metaphase is very distinctive because this is where you see all the chromosomes line up in relatively straight line in the center of the cell, and you can see the mitotic spindle fibers attached to the chromosomes. Anaphase is also distinctive. This is when those 2 sets of chromosomes split. And so what you see are 2 relative lines of chromosomes as they are being pulled by the mitotic spindles to the ends of the cell. The last stage is telophase, and telophase is exactly the opposite of prophase. So in telophase, the chromosomes are going to start unwinding, the nuclear envelope is going to start reforming, and you're going to see the spindle fibers disappearing. Experiment 1, onion root tip. If we were in class, you would have used a microscope to look at cells in an onion root tip and identify different cells in the different stages of mitosis. But because we're not in class, we have found this site which takes the onion root tip and uses a virtual microscope to look at it. When you go to that site, this is what you're going to see. That's the structure of an onion root tip, and you can use that website to get a closer view of the different cells, and you can move around this onion root tip. When you are looking for cells that are in active mitosis, you'll want to look around this area. That's where you will find cells in the different stages of mitosis. Up here in this area, they're not dividing as much, so you are less likely to find the cells there. But go to that site, magnify this area, and look around in that area until you find cells showing the different stages. Then on your lab, in the circles that are on your lab report, you are going to draw a cell in each stage. When you draw those cells, remember that you need to label on your drawings the cell wall, the chromosomes, and if possible, the nuclear membrane, the spindle fibers, and the cell plate. And remember to label your drawings because that's where most of the points are taken off on this part of the lab. Experiment 2 is very similar, except this time you're looking at fish Blatula cells, so these are animal cells. And again, we found a website which has a virtual microscope, and this is what you will see on that website. And then you can magnify different areas to look for cells that are exhibiting the different stages of mitosis. And again, when you draw these on the lab, make sure that you label the cell membrane, the chromosomes, the nuclear membrane, and the spindle fibers if they are visible. The second part of the lab involves biotechniques such as PCR and electrophoresis. PCR stands for Polymerase chain reaction, and this is a biotechnique used to copy DNA. So if you have one fragment of DNA, you can put that fragment of DNA through a PCR reaction, and as it goes through that PCR reaction, you will have identical copies made of that piece of DNA. Another biotech technique is gel electrophoresis, and gel electrophoresis is used to separate DNA by size. So if you have a sample of DNA that has fragments in it of different lengths, you would load that sample into a well in the gel, so this is the agarose gel. Then you would run electricity through the gel to move the DNA down the gel, and this would separate out the DNA by size. Larger fragments move more slowly, so they would remain closer to the well. The smaller fragments move more quickly, so the smaller they are, the further away they would move from the gel. This is an actual picture of an electrophoresis gel. DNA was loaded into these wells, so these are the wells, you can just barely see them. Electricity caused the DNA to move from the top to the bottom, and the DNA was separated by size. So every band represents a group of DNA of the same size. The further away they move from the well, the shorter they are. So these are some measurements here. If DNA moved to this level, they're 44 base pairs long. If they moved here, they were 221 base pairs long. And up here on this side, it shows the longest fragment of 1,857 base pairs long. In class, you actually would have been extracting your DNA from your own cheek cells, but since we're not in class, you'll have to watch a video on how to do it. We have found a video. It's located at this link, which is also in your lab report, and that is what the video looks like. The video doesn't have any sound, but all the information is written there, and you will find everything you need to answer the questions on the lab report. Same thing for PCR. We have found a link to a video about PCR, and again, there isn't any sound, but you will find all the information you need to answer the questions in the lab report. And the last link is for a video on electrophoresis. So go to that link. Again, it's in your lab report, watch the video, and you can answer the questions in the lab.